So coming down the home stretch, we're midway through Tuesday afternoon, second full day of the investigators meeting, and Peng Jin from Emory talked about a new kind of drug screen that he's developed. He basically noticed that if you isolate stem cells from the knockout mouse, that Fragile X stem cells just proliferate more than the stem cells from a regular mouse. So he's able to use this as the basis of a drug screen, put a bunch of cells in little dishes with multiple wells and expose different cells to different drugs and see which ones normalize when they're exposed to a particular drug. He's already isolated quite a few hits from this approach, and using this kind of strategy, these so-called high-throughput drug screens, you can actually identify drugs that have a specific effect on Fragile X without having to know precisely why they have that specific effects. And most importantly, you can get these so-called libraries of available compounds. So you can order up 2,000 available drugs and see if any of them have effects on the Fragile X mouse or cells from the Fragile X mouse. The problem with this kind of study is that it tends to generate a lot of false positives as well as some false negatives, and so you need to go back and actually validate the results. But they're at that stage now where they can start to follow up on some of these hits, and who knows, this is one way to actually come up with potential available treatments for Fragile X in an area that you just might not have thought to look. And it can all be done fairly quickly and at a reasonably low cost. So we're keeping our fingers crossed. It looks like it's going well so far, and hoping we get something out of that. After that, we had some more breakout sessions, and the high points from the breakout sessions. Carol Westmark talked about her very interesting therapeutic approach. As I mentioned previously, she's found that there's too much of this amyloid precursor protein, APP, in Fragile X. And so certain Alzheimer's therapies have experimented with the use of antibodies to APP as a way of just treating Alzheimer's. And so they got a hold of some antibodies to APP and tried injecting them into the Fragile X knockout mouse, and found that indeed they were able to rescue some aspects of the phenotype with this what's called passive immunization. It's important because this is a treatment strategy that's being developed for Alzheimer's, and it may be available in the future. It's not something that we have the money or the influence to develop specifically for Fragile X, but as this proceeds along for Alzheimer's, it could be quite useful. And more importantly, there are other strategies being developed for Alzheimer's that could be useful in Fragile X. So it gets back to this notion of repurposing. So if something is in development for Alzheimer's, and a drug company spent millions and millions of dollars developing it, and then it turns out it doesn't work quite as well as they had hoped in Alzheimer's, we can always approach them at that point and say, hey, we have something else you could try. So that's pretty significant, and I think that's an important potential treatment strategy in Fragile X. And more importantly, it also alerts the Alzheimer's folks to the existence of Fragile X, and allows us to draw on a large pool of talent to get new people into the field. And some of the other talks, another Fraxa fundee, Letitia Davidovic from France, talked about metabonomic profiling of the Fragile X brain. And we've talked about genomics, we've talked about proteomics, but in this case she's actually looking at the metabolic effects of the absence of the Fragile X protein and what that does to a cell and how it affects it. Early on in this kind of study, but she's coming up with some very interesting results, and a lot of the other folks took note of those results as well. There are definitely differences in how the machinery of the cell works in Fragile X. Karen O'Malley from Washington University in St. Louis has been working on a very interesting topic that she presented in the breakout sessions, talking about the role of mGluR5 that's not actually on the cell membrane. It's inside the cell. And it's always been known that receptors get internalized and they're kind of held in reserve inside the cell, and everybody always just assumed that they were just inactive and just being stored there, not doing anything. Well, it turns out that maybe they are doing something. Not a lot of people are working on this, but Karen's lab has found that mGluR5 inside the cell is doing some important things, and that there may be specific differences in how the mGluR5 in the cell is operating in Fragile X versus normal cells. So it's a critical area that we're trying not to overlook. And as we come to the end of the day, we adjourn to have cocktails along with poster sessions where some of the younger researchers were able to show some of their work and maybe provide a little bit more detail on some of the things that were discussed during the plenary sessions. And then moving into the final day, we had some discussions about the role of small RNAs. So you have not DNA, but the DNA gets transcribed into RNA. Now usually RNA we think of as being translated into protein, but some RNAs don't get translated into proteins. They actually do things on their own. They actually have some regulatory functions in the cell. And this is why biologists were somewhat surprised to find out that there are fewer genes in the human genome than they had thought. One reason is that some of those functions are being served by these untranslated RNAs. Or to flip that around, some of the RNAs are just not being translated, they're being used as RNA, not as protein. And so it turns out that there are lots of RNAs that function like proteins, and including one, Henry Teach talked about this BC1 and BC200 RNAs, which have a similar function to the function of Fragile X protein. So these RNAs are repressing protein translation in dendrites in a very similar fashion to what the Fragile X protein does. There's also a great deal of discussion about the mRNAs that are bound by the Fragile X protein and regulated by the Fragile X protein, because that's a critical area of understanding. Unfortunately, even though a lot of work has been done in this area, a lot of the analysis of the data has been done retrospectively. So when somebody comes up with a target that they're interested in, they just go back and look at the list of things that are bound by FMRP, but they don't really proactively go out and try to understand these things. And it's just so much information, it's really hard for a lot of people to cope with. And to end up the meeting, Miklos Toth actually presented some very interesting data that showed, at least in mice, that there's an effect of the maternal genome on the effect of the infant. So you can have wild type mice, pups, are more hyperactive if they have a Fragile X mother than if they have a wild type mother. Even though the pups themselves don't have Fragile X, they don't have any pre-mutations or anything like that. None of that's happening in mice. They're perfectly normal mice, but if their mother was Fragile X, then they are actually more active than their litter mates. So, interesting epigenetic effects, as we call them. So after that, we had a little bit of discussion. It was pretty clear that everybody was really psyched. I think the timing for the meeting was really fantastic because the New York Times article got everybody really, really interested. We had a huge number of pharma people who were paying very close attention. Obviously, we were checking in on them from time to time to see how things were going, and it's pretty clear that they were very psyched about what they heard. And so we all left with a great anticipation for the next meeting, but we've also had a flurry of activity following that meeting. Now the time is right to try to follow up on what we did, form collaborations, get things moving, not just planning the next meeting, but actually planning research to follow up on what happened at the meeting. So that's where we're at right now. We'll hope to keep you posted in the future. Thanks for watching. I hope this has been helpful for you, and I hope to do more videos in the near future. Bye-bye.